Hello, welcome to Biology, the scientific study of life. Reproductive cloning of mammals. The evidence that all cells in an organism have the same DNA also comes from experiments with mammals. Researchers have long been able to clone mammals using nuclear cells from a variety of early embryos, but it was not known whether a nucleus from a fully differentiated cell could be reprogrammed to be totipotent. However, in 1997, Scottish researchers captured newspaper headlines when they announced the birth of Dolly, a lamb cloned from an adult sheep by nuclear transplantation from a differentiated cell. Notice this figure. These researchers achieved the necessary differentiation of donor nuclei by culturing mammary cells in a nutrient-poor medium. The researchers then fused these cells with sheep egg cells whose nuclei had been removed. The resulting diploid cells divided to form early embryos, which were implanted into surrogate mothers. One of several hundred implanted embryos successfully completed normal development and Dolly was born. Notice that this is Dolly. Later analysis showed that Dolly's chromosomal DNA was indeed identical to that of the nucleus donor. Her mitochondrial DNA came from the egg cell donor, as expected. In 1933, at age 6, Dolly suffered complications from a lung disease usually seen in much older sheep and was euthanized. Dolly's premature death, as well as her arthritic condition, led to speculation that her cells were older than those of a normal sheep, possibly reflecting incomplete reprogramming of the original transplanted nucleus. Since 1997, cloning has also been demonstrated in numerous other mammals, including mice, cats, cows, horses, and pigs. In most cases, the goal has been to produce new individuals. This is known as reproductive cloning. We have already learned much of interest from such experiments. For example, cloned animals of the same species do not always look or behave identically. In a herd of cows cloned from the same cell line, certain cows are dominant and others are more submissive. Another example is the first cloned cat named Copycat. She has a calico coat like her single female parent, but the color and pattern are different due to a random X chromosome inactivation, which is a normal occurrence during embryonic development. Clearly, environmental influences and random phenomena can play a significant role during development. The six-fold cloning of various mammals has heightened speculation about the cloning of humans. In early 2024, Southern Korean researchers reported success in the first step of reproductive cloning of humans. In this work, nuclei from differentiated human cells were transplanted into unfertilized eggs from which the nuclei had been removed. These eggs were stimulated to divide, and some reached the blastocyte stage in early embryonic stage, similar to the blastola stage in figure 1. This figure, notice that, like this. Oh, voila. Although the embryos were not allowed to develop beyond the blastocyst stage, the work of these researchers bring us one step closer to the possibility of human reproductive cloning, which raises unprecedented ethical issues. However, problems associated with the cloning process have bought us a little more time for thought. Problems associated with animal cloning. In most nuclear transplantation studies, thus far, only a small percentage of cloned embryos develop normally to birth. And like Dolly, many cloned animals exhibit various defects. Cloned mice, for instance, are prone to obesity, pneumonia, liver failure, and premature death. Scientists believe that even cloned animals that appear normal are likely to have subtle defects. In recent years, we have begun to learn possible reasons underlying the low efficiency of cloning and the high incidence of abnormalities. In the nucleus of fully differentiated cells, a small subset of genes is turned on and expression of the rest is repressed. This regulation often is the result of epigenetic changes in chromatin, such as acylation of histones or methylation of DNA. Many of these changes must be reversed in the nucleus from a donor animal in order for genes to be expressed or repressed appropriately for early stages of development. Researchers have found that the DNA in embryonic cells from cloned embryos, like that of differentiated cells, often has more methyl groups than does the DNA in equivalent cells from uncloned embryos of the same species. This finding suggests that the reprogramming of donor nuclei is not always complete because DNA methylation helps regulate gene expression. Misplaced methyl groups in the DNA of donor nuclei may interfere with the pattern of gene expression necessary for normal embryonic development. Back to a research method, reproductive cloning of a mammal by nuclear transplantation. Application. This method is used to produce cloned animals whose nuclear genes are identical to the donor animal, supplying the nucleus. Notice also a technique shown here is the procedure used to produce Dolly, the first reported case of a mammal cloned using the nucleus of a differentiated cell, mammary cell donor and egg cell donor. Notice that cell from ovary, notice here a nucleus removed, and this cultured mammary cells are semi-starved, arresting the cell cycle and causing differentiation. Notice cell fused and a nucleus from mammary cell and grown in culture, early embryo and implanted in uterus of a third sheep, surrogate mother, and embryonic development. Lamb Dolly genetically identical to mammary cell donor. The results: the cloned animal is identical in appearance and genetic makeup to the donor animal supplying the nucleus, but differs from the egg cell donor and surrogate mother. Okay, thank